Our first speaker today, Dr. Lawrence Goldstein, is celebrating his 21st anniversary here at the university, and pardon me for mentioning 21. And one of the interesting things that I discovered about Dr. Goldstein's research is it's not just advocacy in the sciences, but it's advocacy in the political arena. It's advocacy in the public arena to engage the public and help the public understand the importance of the research that is going on at institutions. Dr. Goldstein has testified before the Senate in the days of controversy regarding stem cell research in 1999. Dr. Goldstein went before the Senate committee and explained the importance of maintaining funding in public institutions as well as private institutions, recognizing the talent that exists in our public institutions here. For my benefit, Dr. Goldstein is also an author of the Stem Cells for Dummies book. And reading a review of the book, it was well received. It was not determined to be overly ambitious, although all of our all-star researchers are certainly overly ambitious. And it was reviewed to be an excellent guide for someone having an answer to, or needing an answer to a specific question. This research is sure to lead towards innovations that will help many treatments that are personal to so many people, from Alzheimer's and other degenerative diseases. And it is with great pleasure that I introduce Dr. Goldstein today.
This is something of an anniversary. 21 years is interesting. So I guess I'm old enough to drink at the University of California now, finally. But the other thing that struck me as I was listening to Bob Dines is that when I first moved to UCSD from Harvard University, it was also an east to west odyssey. But interestingly, I first knew Bob as a scientific colleague when I arrived at UCSD before he became an administrator, but we think you're still part of the team anyway. That's good. And it was meeting him as a physicist that was actually very important to me, because as a newly arrived molecular biologist at UCSD, not only did I already have built-in interactions with folks like Nick Spitzer, who was a well-known neuroscientist at the time, but very soon after my arrival, I was interacting with physicists and engineers and physical chemists and people in other disciplines, and it led to a great many collaborations. Some of them led to the formation of a company that I still hold dear, and it's part of what makes this institution, UC San Diego, and the surrounding institutions so incredibly interesting to work at, is the degree of intellectual interactions outside one's discipline in a very freewheeling sort of way that just didn't exist on the East Coast, and doesn't exist at most institutions, to be honest. And so what I'll do, let's see if I'm going to get this right. How does this work here? Oh, there we go. Okay, good. All right, so that's me. And so what I'll talk about is stem cell science. And what I'll give you is sort of a three-part story. The first part is just to remind you of the basic background of how stem cell science works. It'll be a little more advanced than Stem Cells for Dummies, so I will not go back to full lay language. You guys are far more sophisticated than that. But I do want to make sure that we define our terms and agree on what we're talking about. I'll be returning to Bob Dines during that part of the presentation, as you'll see, so we'll keep him on the edge of his seat. And then I'll talk a little bit about how my own lab has been using stem cell technology to probe and to develop therapies for really terrible diseases, Alzheimer's disease and Lou Gehrig's disease, ALS. And then I'll close at the end telling you how we as an institution at UCSD are riding the next wave of stem cell science into the development of useful therapies as a result of the formation of a new program at UCSD with the help of our colleague and friend, Denny Sanford. So I have to start with the usual financial disclosures. You folks are professionals. You know how to deal with this kind of information. I'm subject to personal financial gain. I have biases because of trials and other activities at the university. My friend Irv Weissman sometimes says things like, well, from here on out, you shouldn't believe anything I say because I'm so biased by my conflicts of interest that I couldn't possibly be objective. So I'll leave that to you to decide. So. And the background for this really does start about 15 years ago in the U.S. Senate, because up until the announcement by Jamie Thompson's group in Wisconsin of human embryonic stem cells, I had no idea that I would be working on humans at any time in the near future. And the reason I was asked to testify in this hearing in the Senate, in a subcommittee chaired by then Senator Arnold Specter, was because my lab and I had worked on mouse embryonic stem cells for a number of years, and therefore I knew something about the technology and what one might do with it in the human. In the first hearing that I testified in, one of the folks I testified with was Christopher Reeve, then an actor, and as many of you know, also a victim of a horseback riding accident. Christopher had a break in the cervical region of his spinal cord, and as a result was basically paralyzed from the neck down with very, very minimal function. Chris dedicated the rest of his life to the support of research to develop stem cells and any other potential technologies into useful therapies for spinal cord injury, which is a very common injury in our society. But Chris also had a way of capturing what I would call inspirational statements in very plain language. And an example of that is shown here, where he talked about what really drove him was the notion that medical research and then translation, RD&D as Bob puts it, really had the potential to take words like impossible and unsolvable, which was the state of spinal cord injury therapy 15 years ago, to the realm of actually changing the ability to treat and actually reverse and eliminate some of the symptoms as a result of these injuries. And as you'll see at the close, while we're a few years too late for Christopher, we are about to launch spinal cord injury stem cell trials at UCSD. Other institutions and companies around the world have already begun, and actually it's turning into a bit of a hot competition to see which of these therapies will work and then work best for the treatment of this sort of injury. It's really quite remarkable what's happening. Now, let's start at the beginning, and that is with the question of what are stem cells? What do I mean when I say stem cell? There's a lot of misinterpretation of the term in the public and business domain. And then what do we think about how they can help? I've got to keep an eye on the clock here. Okay, good. And so let me give you a quick definition. So if we go back to Bob, this is the part where Bob is the audience participation. If we go back to Bob, and should I make him stand up? Yeah, what the heck? Okay, Bob. So we're going to stand up. Oh, come on. Yeah, you. So Bob's going to stand up, and we're going to look at Bob, okay? So we're going to make him the center of our attention. Not a pretty sight. Not a pretty sight. No, actually, he's a very handsome gentleman, aging very gracefully. But that's not the point I'm going to make. So what you'll see is that, we learned in high school biology that cells grow by making more cells by cell division. And we learned that those two daughter cells from a cell division were identical, right? Okay, that's what I call the biggest lie in biology, and you guys already know why. See, if we look at Bob, Bob is not just an accumulation of identical cells, right? He's got hair cells, eye cells, skin cells, muscle cells, plenty of those, heart, lung, liver. Okay, I'm done with you for the moment. So here's the demonstration of why that basic dogma can't possibly be correct in its simplest form. And part of the resolution of that dogma, not all of it, but part, is that there are special cells in our bodies that during development, when they divide, the two daughter cells are not identical. That's important, because one of the cells remains what we call a stem cell, and the other cell, if it gets the right kinds of signals, can go off and become a hair cell or a heart cell or a liver cell in a controlled way. So ultimately, a stem cell is a cell that we define as one that can divide to make more of itself, as well as its progeny responding to biochemical signals to make specialized types of cells, such as though we find in an aging, gracefully gentleman, such as Bob and myself, I'm afraid to admit. So that's actually important, because that means, if you think about it, that this is a very important enabling technology in biology and biomedicine. And so I'll return to a second example, which is a film that was made in the 60s with Dustin Hoffman as a character who was trying to figure out what his career path was going to be. And he received some very important advice at a party he attended just after he graduated from college. I'm sure you folks all remember the career advice Dustin Hoffman received. Plastics, there you go, that's your cue. Okay, very good, you're paying attention. And in the 60s, that was the right career advice, because plastics were the future of manufacturing in many ways. We were no longer going to use steel and wood for everything we built, we were going to use plastics. And the result of that you can see in this building, in this room. Many things we used to build out of natural materials, we now build out of plastics. It was great career advice. Well, in 2014, the advice I would give is biological plastics. And what I mean by that are stem cells, because stem cells give us the ability to control the development of different sorts of biological materials, to develop cells that might replace those lost in disease, or to give us a way of studying human biology in the lab using human stem cells that we were never able to do in the past. And so the coming era is one of biological plastics, where we will learn how to use these cells to tackle problems in disease, to develop new bits and pieces of organs, and I suspect to do things that none of us is smart enough to anticipate. That's the beauty of enabling technology. When smart people come into contact with enabling technology, they think of things to do with it that you never would have imagined, right? Look at recombinant DNA and its use in forensics. You never would have predicted that in the 70s, right? So it's a totally amazing thing that happens when useful enabling technology gets into the hands of largely smart people at public and private institutions. And so stem cell technology, as you'll see, is giving us a way to tackle problems that have, as Chris Reeve would have said, been previously intractable. And in the case of Alzheimer's disease, ALS, and spinal cord injury, I'll give you examples of how we're doing that now. Now, one of the confusions in the world is that, first of all, there are actually many different sources of stem cells. You can get them out of cord blood, you can get them out of amniotic fluid, there are so-called embryonic stem cells, and there are now reprogrammed stem cells, a technology that won Shinya Yamanaka the Nobel Prize a couple of years ago. And the message I'll just leave you with as tech transfer and business development professionals is that when somebody says stem cell, the first question you should ask is what kind? Because just as all of the different tools in a toolbox have different uses and different sensible uses, you wouldn't use a screwdriver to hammer a nail, for example, although that's a creative use of the technology, I suppose. In fact, knowing what the different types of cells are when somebody approaches you with some great idea will help you understand how best to develop it and think about it. I'll note, for example, that there are clinics all over the county and the world that will propose to take stem cells from your fat and use them to treat every disease known to mankind. This is actually an example of fraud, it turns out, because the cells that they claim to be generating, so-called mesenchymal stem cells, we now know as a result of scientific presentations I heard in the past few days at the International Society for Stem Cell Research meeting. The claim is that these are so-called mesenchymal stem cells, and the fact is whatever these folks are getting out of fat is not. But if you look, for example, in bone marrow, you can find cells that legitimately make all of the cells of the blood system and the immune system. Embryonic stem cells, which come from early frozen embryos left over after IVF treatment, those cells can be grown to quite large quantities and can make any cell type that we find in an adult human, such as Bob or myself. And reprogrammed stem cells have many of those same kinds of properties and give rise to all sorts of different kinds of uses, as I'll illustrate now. And so the second part of what I want to talk about is how we and others are taking this kind of stem cell technology, where we can grow large quantities of cells and direct them to generate different sorts of adult cells, and how we're using them to tackle, in my case in particular, problems in neurodegenerative disease such as Alzheimer's, Parkinson's, ALS, and others. And of course, the background, as you know, is that we currently have no drugs, no useful therapies that change the course of any of these disorders. We have drugs that help with some of the symptoms, but there is an incredible and pressing need for drugs that will slow the development of these diseases in a person who has them. It's a terrible need, actually. And so one of the approaches that we and others have used that takes advantage of stem cell technology is so-called disease-in-a-dish technology to tackle problems such as Alzheimer's disease or others. And the basic notion is, if we return to Bob, if I took a sample of Bob's skin cells with a small biopsy, I could then generate so-called reprogrammed stem cells that have Bob's unique genome, his unique DNA, in stem cells that I could then use to make neurons, brain cells, other types of brain cells called astrocytes, and I could study how those cells interacted in a dish and use them to test for drugs that would perhaps reverse Bob's personal set of disease and disorders. And I'll come back to that in a moment. The particular case that we've been using this for that's illustrative of this technology is the case of Alzheimer's disease, where we know a great deal about the development of pathology and the loss of connections between brain cells before they die, but where we don't really know what the basis biochemically and cellularly is for both the formation of the pathologies in most people who develop disease, and then in particular, how some of the detailed biochemistry of this protein that I'm pointing to right there, called amyloid precursor protein, which we know is part of the development. Can you see those? Oh, I can see this little thing and you can't. Well, too bad. Down at the bottom there, we know that protein is integral to the development of pathology and disease, but we don't know exactly how. And so one of the things we've done in my lab, and this is work from a really smart graduate student of mine, Mason Israel, was to take a number of individuals who either had rare hereditary forms of Alzheimer's disease or who had what we call sporadic Alzheimer's disease. You guys know that sporadic disease is code for we don't know what the heck caused it. It's the kind of disease most of us will develop. And then some patients from our Alzheimer's Center who are controls. And we reprogrammed them using this technology and used them to develop, as shown here, purified neurons in a dish that either had the genetic changes that caused hereditary Alzheimer's disease or the genomes, the DNA of people who developed sporadic disease, where we know there's a big hereditary contribution, but we don't know how or why, and also controls. And then compared the properties of those neurons carrying all these different genomes biochemically in the lab. And what Mason learned was that we could recapitulate in a dish, amazingly, some of the abnormal biochemistry that we see in early stages of Alzheimer's disease. And that's what this slide illustrates. And without a pointer, it's going to be a little hard for me to point to it. But if you just look at the far right graph there, you'll see that the two rightmost bars in that graph are high. The two leftmost bars are low. That's controls compared to cells from people, cells with the genomes of people who had hereditary disease. And you can see that there's a big difference in one of the downstream biochemical changes that we see in these cells and in the development of this disease. And what's remarkable about this ability to both generate cells in a dish that carry the DNA of people with particular forms of disease are that we can see these very simple early biochemical changes, but that using other genetic tricks, we can elucidate what the biochemical steps are in the lab in these pathways. That's a really interesting problem, it turns out. But even more important, using this very simple kind of assay, we've started testing FDA-approved drugs with folks at the Sanford Medical Institute in their Prebys Chemical Genomics Center. We're looking for FDA-approved drugs that change these biochemical features of disease in a dish. And remarkably, we have already, even in a few months of work, it took a while to get this to work properly, don't underestimate the difficulty here, but once we've gotten this to work properly, we have found a number of really interesting hits that you wouldn't have found by drug testing by any of the means that pharmaceutical industries conventionally use. And so we think that these can, very quickly after we finish characterizing them and figuring out what their mechanisms are, get them into early stage clinical trials to begin testing, will these really be efficacious for actual disease in people, leapfrogging off this very relatively simple cellular set of assays that we can pull off in the lab. So that's one example of so-called, oh, thank you, Nick. Wow. Yes, that's the graph right there. You just remembered you had to point it? Somebody gave it to you as a Christmas present, whatever. Thank you. So it's a remarkable technology, and that's one example of it. Now, the second way this technology is turning out to be incredibly useful is to dissect how human genetic variation predisposes to common, sporadic forms of disease. And that is this ability to capture in stem cells the genomes of individual humans. And that's important because if I want to study neurons, brain cells, that have the DNA of individual people, if we return to Bob again, if I were to say, Bob, you want to come up to the lab this afternoon, we'll get the drill out, we'll drill a little hole, scoop out some of your brain, Bob's response to this would be, hell no. Hell no, that's right. I've gotten that response a lot. I did get one yes once. That person should have their head examined, and we were about to do that. That's right. So we can't get Bob's neurons in a dish without doing this method of taking skin cells, reprogramming them, and then we can generate Bob's brain cells in a dish. We could actually generate Nick's brain cells in a dish. We could generate a mixture and see how they talk to each other. This technology is incredible for studying cell interactions. And so we've recently done that with Craig Venter. So Craig, you know, completely sequenced his own genome with very high accuracy and fidelity, and so as a result, he's one of the rare genomes where we know all of his risk factors for the development of disease. And so Craig let us biopsy him, and then we used the so-called Magic 4 retroviruses to reprogram his cells into stem cells, and folks in my lab have now literally been studying Craig's brain cells in a dish. And not only are they aggressive and megalomaniac, but they actually have things to teach us about the variants that predispose him to disease. And in fact, we've now sampled a large number of individuals who have different variants in their genomes. And we've discovered recently at least how one of the genetic variants that is known to predispose to disease causes biochemical changes in brain cells that have that variant. It's taught us a great deal about how we might drug that pathway. And it's also told us how to go about stratifying clinical trials potentially, so that people who have different variants, we would actually make different predictions about how they'll respond to certain kinds of drugs. And we just wouldn't have gotten that kind of insight without this ability to probe how variation predisposes in a dish. So the final example is ALS, Lou Gehrig's disease, where we know that in the development of this disease, what happens is that people progressively lose their motor neurons that control movement of their muscles, and ultimately, after all of those motor neurons have died, these people are completely paralyzed. They can't breathe, they can't swallow. It's really terrible. Some of them go on ventilators, feeding tubes, what have you, but it's a very devastating disease. Usually, average lifespan after diagnosis is 3 to 5 years, and there is no drug that really changes that basic timeline very much, and some people die within a year. It's a terrible, terrible disorder. So the question is how to go about using this kind of technology to treat Lou Gehrig's disease successfully. And there are a number of labs around the US and the world that are using the so-called disease in a dish approach to try to devise drugs to treat Lou Gehrig's disease. We've taken a slightly different approach because as a result of work we've done in the past decade with my colleague Don Cleveland at UCSD, we know that even though it is these long motor neurons that are dying, other cells in the spinal cord called astrocytes play a very important role in potentially speeding the death of motor neurons or slowing the death of motor neurons if we manipulate the astrocytes directly. And so that has led to a project that I currently lead, where what we're trying to do is to start with human embryonic stem cells develop in the lab by taking advantage of these cells' ability to differentiate and grow in vitro. We generate precursors to neurons and glia, astrocytes, in a dish, and we know that if we transplant these into experimental animals, not yet the human, but soon, if you're a rat or a pig, these cells will differentiate into large numbers of functional astrocytes in the spinal cord of these transplanted animals. And so the hope is that we can actually get these cells into clinical trials in the next few years in ALS patients to test whether we can slow or reverse the course of that disease. And there are other groups out there using different types of neuronal precursor cells, and the point is it's really only through clinical trials that we'll be able to understand which types of cells will slow or, we hope, at some point reverse the course of this disease. Now, as Bob mentioned, and this is the final part that I'll hit briefly, is to talk about how we and others are taking this kind of science and using it to develop appropriate and rapid clinical development programs at the University of California and elsewhere. Bob mentioned that guys like me who spent our lives at other institutions prior to moving to UCSD, such as Harvard, learned that most of the academic world is built as silos, so that you're in a department, for example, the Department of Corn, and if you wanted to talk to somebody in the Department of Wheat or soybeans, you were pretty much out of luck. But of course, what I told you at the beginning is already when I arrived as a biologist, I was quickly speaking and interacting with really smart physicists such as Bob, engineers such as Shu Chen, and a great deal of other individuals. And of course, that's the point of how we do things in San Diego, and it's what led to the development of the Sanford Consortium for Regenerative Medicine, which in a sense is a set of bridges between institutions and disciplines in the area of stem cell research with the goal of developing good ideas that might then be translated into clinical medicine. And of course, the next step in this progression, as you may have recently heard, is that, again, our friend Denny Sanford stepped in with a large gift to UCSD to establish the so-called Sanford Stem Cell Clinical Center, which is really dedicated to accelerating stem cell-based clinical trials in humans. It really is the only way, as you know, to develop effective treatments is at some point you have to do research with human subjects. And so the major goal of the center is to combine ethics, education, and a hell of a lot of collaboration in science and medicine to bring new therapies to patients over the next decade or so. It's a 10-year and more project. My job is to make it a sustainable project at the end of 10 years. I have a lot of work coming up. And one of the basic means of doing this is, as you guys know, one of the biggest obstacles to development of good scientific ideas and discoveries into useful therapies, we have to go through the so-called valley of death, where there really is a dearth of risk capital in the world and a variety of other problems. And so part of the goal of the Sanford Stem Cell Clinical Center, if you will, is to live in the valley of death, and in a sense to function, as you'll see here, as a big funnel. And you're going to ask, what the heck do you mean by a big funnel? And really the idea is here, and that is to find ways to take and accelerate scientific discoveries from our partner institutions in San Diego and elsewhere as needed, and to bring them as rapidly as possible to experimental trials and hopefully the development of treatments by taking advantage of this unique gift from Mr. Sanford. One example of that is actually shown here, where because of Mr. Sanford's gift, we have been able to accelerate the development of a phase 1 clinical trial for spinal cord injury using cells that come from a company called NeuralStem. These are fetal stem cells that will make neurons and astrocytes and other types of glia following transplantation. And what's remarkable is shown at the bottom, which is if you're a rat, we don't yet know about humans, but if you are a rat with a spinal cord injury and these cells get transplanted to the site of injury, they will grow in a way that adult cells just don't seem to. Long fibrous connections in both directions across the injury site in the injured rat. And these cells appear to establish contacts with cells both above and below the site of the injury. And again, if you're a rat, there is large restoration, though not complete cure, of function. And so we cannot continue experimenting on rats forever, and as a result, we will begin clinical research with humans with these cells. We expect to do 8 patients in the next 6 to 8 months to see how humans respond to this kind of transplantation. We are hopeful, but as you know, clinical research in humans, we are not just big rats, you may have noticed, and so the response to the cells may be different, but it's a really important next step. So let me close there. I've used up my time. I hope that you've gotten a sense of the kind of excitement that is happening around here in San Diego in these fields, and maybe to get a taste of how we intend to accelerate their application in human patients. Thank you. Okay, so what happens now? Do I answer questions, or do I give up the microphone? Or
does anyone have any questions for Dr. Goldstein?
You may have read my book, so then there'll be nothing out there to tell. Yeah.
So as your work progresses with Alzheimer's, will the results actually translate to other diseases?
So the question is, as we make progress on Alzheimer's disease, will the results be applicable to other diseases? And I think the answer is yes. So all neurodegenerative diseases share a couple of features, it turns out. One of them is the early loss of connections between neurons, and the other is some of the pathologies that develop overlap greatly between different diseases. And in fact, some of us joke that the different diseases are categorized with names because the neurologists need to know which branch of the hospital to refer them to. And in fact, we know in some cases that similar genetic defects can give rise to either autism, schizophrenia, or bipolar disease, depending on which individual has them. And we know there's a great deal of overlap, for example, between Alzheimer's and Parkinson's. So we suspect that there will ultimately be a great deal of so-called off-label use, and that the field may evolve in the way that cancer has evolved, where you're not categorized necessarily by which part of your body has cancer, prostate or breast or liver or what have you, but you'll be categorized by the genetic change that has driven the development of aggressively dividing and migrating cells. And that's a very important realization being capitalized on now at the Moores Cancer Center, where, as some of you may have heard, a company of Craig Ventures that I freely disclose, serve on the board of, they're going to sequence every cancer patient who walks through the door to begin understanding how their genomes predispose. And so I think it's a really interesting set of projects, but I think this will be similar in the neurodegeneratives as well. Yeah.
So Larry, so we've got in San Diego now world-class research and development on stem cells. We've now got world-class clinical. The other part of that equation, the kind of the third leg, is the manufacturing, the ability to really manufacture these in a GMP fashion. How do you see us getting into that area? It's a loaded question.
It is a very loaded question, Paul. So as Paul knows, there is an ongoing study in San Diego to ask whether we should construct a manufacturing center here in San Diego for the transition of stem cell therapies from what will be small phase 1 trials, where one doesn't need to manufacture huge quantities, to phase 2 and phase 3 trials, where you potentially need to treat hundreds or thousands of individuals. And so whether or not we should build one here in San Diego is a great question. It's an ongoing debate. Certainly the governor of Texas has thrown down the gauntlet as to whether he thinks it should be in Texas or not. I think we should never let Texas get a leg up on us here in California and San Diego, but we have to make sure that if we use civic support to build something, that there is adequate levels of need to support and sustain an operation like this in the coming years. So it's a really hard kind of problem, but you study it and then we'll come up with some sort of recommendation. Great, thanks.
All right, well, special thanks to Dr. Goldstein.